Aplacophora mollusks. I realize this isn't exactly a household term. Briefly, these are worm-shaped marine mollusks that are lacking a shell but covered with these calcareous structures we call sclerites, which is kind of a catch-all term for spines or scales or other sort of epidermal calcareous structures they have. There's two major groups, the caudofoveates, which are shown in A through C here. And so these are basically honorary worms. They're burrowing, they completely lack a foot. We think they eat foraminiferans, although we know that from some genomic data that some also feed on annelids. There's about 130 described species, but probably on the order of 10-fold more actual biological species out there, if we take a guess. And then the other clade is the selenogasters, which are also called neomorpha, which is D through H here. And so these animals do have a foot, so they're kind of like a scaly or spiny slug. They are epibenthic. You can find them crawling on the surface, or the meiofauna taxonomy, of course, between grains of sand. There's also a lot of taxa that you can find wrapped around corals or gorgonians, which they tend to feed on. This is a more diverse group. There's about 350 species, but again, we know that the diversity of this group is really underestimated. And like many groups of meiofauna or small marine invertebrates, this is a really understudied group. We have a lot to learn about them. So why do I care so much about this?
Just one second, Kevin.
Yes.
Can you speak slowly, because there's many people complaining about the sound be a bit dodgy.
Okay.
But I think there's some internet problems around. And then just go a little bit slower, that will be easier for the people to get it, okay?
Sure. Sorry, I talk fast.
No worries. But
thank you. So the reason I'm really interested in this group is because Aplacophorans, along with chitons, are the sister group to all other mollusks. So they fall out in this clade called Aculiphora, which is sister to the more familiar mollusks like gastropods and bivalves and cephalopods. And so Aplacophorans and chitons are then really important for understanding early molluscan evolution. I think this is a really diverse and interesting and important group. We have everything from colossal squid to microscopic worms that live between grains of sand. And so studying Aplacophorans can help us get a better understanding of this really interesting group. As a brief aside, our lab is also doing work on chitons. And I want to plug my student Rebecca Varney's work on the chitin Acanthopleura granulata. So chitons are a cool model for biomineralization because in one animal we have shells, we have these sclerites. We also make teeth out of magnetite, which is basically as hard as tank armor. And so if you're interested in biomineralization or chitons, she sequenced the genome of this animal, and that's up on bioRxiv now. So back to Aplacophorans and meiofauna. Like a lot of groups of meiofauna, there's not a lot of taxonomic experts on Aplacophorans out there. And unfortunately, in the last few years, we've lost a significant number of experts on the group. And so in particular, Ami Scheltzma, Ludfried von Simonyi-Plaven, and Christopher Shander are three experts, and especially Ami and Ludfried described about a third of the named species of Aplacophorans. And so their loss was a big hit to our community. Now on the upshot, there's a lot of early career researchers working on this group and other people who are still in training. And so I think we have a bright future for Aplacophorans, but it's a very small community. And so despite having this fairly small number of people who are actively working on this group, there's a lot of what we call known unknowns out there. So species that we know are undescribed, but no one's had a chance to describe them. And also a lot of material from various environmental surveys that's accumulated and needs to be looked at, and probably almost certainly has more undescribed species. I'll use Australia as an example of this. There are 10 species described from the entire continental region of Australia, but one set of toes, trawls in the Bass Strait by Ami Scheltzma revealed something like 65 morphospecies in just that area. So we have a lot to learn about probably most of the Aplacophoran fauna of the world. So one reason why there's so many undescribed species and so much material that hasn't even been identified is this is a somewhat challenging group to work with. And I think a lot of meiobenthologists can commiserate with this, and different groups have different challenges. But in particular, these animals are not transparent. And so I'll use this species, Hypomenia sanjuanensis, that Christiana Topp and I described back in 2013 from Friday Harbor, Washington. And I've seen little brown spiny worms that look pretty much just like this off Washington, in Icelandic material, in Antarctica, and probably elsewhere. And even under the SCM, a lot of these animals look very similar. But that's not because they're just simple and boring. When we do histology and look at their internal anatomy, various anatomical structures, in particular foregut glands, their digestive system, and reproductive anatomy, and radula, are really informative into their taxonomy and can help us identify them. But it's not really feasible to do histology on every single specimen just to identify it. We are doing this for species description. I think this is really important for describing new species. And so here's kind of an example of a workflow that my master's student, Maddie McCutcheon, is doing. So she's focusing on New Zealand, which is incredibly understudied with respect to its Aplacophora fauna. We have boxes of specimens kindly provided from NIWA and Museum of New Zealand Te Papa Tongarewa, and she's got dozens of species. This actually isn't one of her species, but this is a nice photo of this genus anyway that she's working with, taken by my postdoc, Carmen Cobo. But so Megan's been doing histology and then this traditional anatomical reconstruction where she's looking through the micrometer, measuring things with the reticle and using graph paper and colored pencils to develop these really nice anatomical reconstructions, which we need to tell some of these things apart. Maddie has several species that look pretty much just like this. And so it's a challenging group to work with without histology, but it's a lot of work to do that, to describe and certainly to identify specimens. And so kind of the state of the field, in my view, is we have a number of unanswered questions and challenges that we need to face. And so first and foremost is how many species of Aplacophorans are there? Christiana Tott in a paper in 2013 estimated there's probably tenfold more, but that's some back of the napkin math, and we really don't have a good sense of how many more we have left to discover, but certainly a lot. How many of the Aplacophorans that we think are one species are actually one species? We have started to find a lot of cryptic species, and I think both of these points so far can hit close to home for many people working on meiofaunal taxa. How can we reduce the need for labor-intensive morphological lab techniques, and I'm referring to histology, for specimen identification? When we describe new species, we want to use histology. We want to have all the information we can. But it's a lot of work to do histology just to identify something. And then, of course, when we do that, we destroy the specimen and can't use it for anything else. So what do we do when we find just one of something? A lot of Aplacophorans are kind of like nudibranchs in that they're somewhat rare. They tend to be picky eaters, and we may just find one of the species. And so do we do histology? Do we do SCN? Do we do a DNA extraction and barcode it? And then finally, as I said, Aplacophorans are kind of in an important phylogenetic position. And so by studying their evolutionary history, I think that can help us better understand the early evolution of Mollusca as a whole. And so to that end, I'm really happy to share that last year I received funding from the US National Science Foundation to help support tackling some of these questions. And so specifically, the goals of my career project are to host a number of meiofauna diversity and taxonomy training workshops, to use an integrative taxonomic approach to try to basically squeeze as much data as we can out of each specimen, and to identify and describe species. We want to make a reference DNA barcode library from expert identified specimens. This is work my collaborator Christiana Tott in Bergen, Norway has already been doing a lot of work on. And so we're hoping to add material from other parts of the world that we're sampling. So that way in the future, non-experts will be able to at least take advantage of sequencing to put meaningful labels on specimens. We want to reconstruct the phylogeny of the group, and I'll talk a little bit about that using a target capture phylogenomic approach. And then finally use that phylogeny to shed a little more light, hopefully, on molluscan evolution as a whole. And so briefly, we have a number of workshops planned. We were hoping to have one this June, but unfortunately that was postponed until next year. That will be at the Smithsonian Marine Station in Fort Pierce, Florida. We also tentatively have one planned for the Dauphin Island Sea Lab in Alabama, Friday Harbor Labs in Washington, and the Hawaii Institute of Marine Biology in Hawaii. Additionally, we have 2 Antarctic research cruises planned. One is planned for November to December of this year, which looks like it's still happening, fingers crossed. And we have another cruise planned for 2022 or 2023 in the Eastern Antarctic, which we're very excited about because there's not much material available from this region for a lot of different taxa. And so for those of you who might have a wish list for these cruises, please let me know. So a little bit of an origin story for Kevin. I really got into meiofauna when I took a course in 2010 at Bocas del Toro, Panama, the Smithsonian Marine Station there. This is a workshop organized, I believe, by Rick Hochberg and John Nornberg, where basically they got a room filled half with world experts on different groups of meiofaunal organisms and half with kind of aspiring invertebrate taxonomists and systematists. And they provided us with plenty of sand and microscopes, and just kind of turned us loose. And I learned a ton. I got to see so many things I've never seen before, but more importantly, I got to network and learn directly from experts and meet a lot of colleagues. And so I think a real testament to the success of these sort of training workshops is the fact that over half the participants of this workshop are now professional invertebrate zoologists or on their way to becoming one. And so we can debate about causation and correlation here, but all of these people are people I'm still in touch with. They're actively doing research in this area, and I think these are really valuable training opportunities. And so it's something I'm really dedicated to and would love to continue after the life of this grant. I know there's been a number of Mio schools and other workshops organized by different groups, and I highly encourage any of the students or early career folks to take advantage of these or to get involved in helping to lead them. And so we'll be advertising these via my Twitter and all over social media when they're coming up, so stay tuned. So I mentioned this integrative taxonomic approach you want to use to try to speed up the identification and description of these animals. And the workflow we've come up with is to continue to take nice photos of all of our specimens using light microscopy. But then simply for identifying specimens, histology just isn't really practical. It's destructive and it's time consuming. And so what we've been doing instead is started using microcomputed tomography or micro-CT, where we reversibly stain specimens with iodine. We basically do X-ray on them. And so you X-ray the specimen along multiple different planes and computationally stitch that together to get a 3D model. And this is non-destructive. We can still get, for example, DNA out of them. If we use iodine, we don't decalcify them. So we could then use them, for example, for SEM. And so in my lab, we have this really wonderful tool. This is a Phenome Pro tabletop SEM. It has a low accelerating voltage and it's a low vacuum SEM. And so what that means is we are able to take specimens that were in alcohol, dry them out, put them directly onto a stub without osmium or critical point drying or anything like that, and get really nice images, at least of the hard parts. So this doesn't work well on soft specimens, but it would work well on anything calcareous or things with the cuticle like nematodes or chinorhynchs. And because it's a low energy, we can then extract DNA from the same specimen. It's not high molecular weight DNA, but it's just fine for barcoding and even like an Illumina library prep. And because the first step is to lyse the tissue away from the cuticle and the sclerites, we can then pellet out the sclerites and make a permanent morphological voucher. And we can do all this from the same specimen. And so if we have just one of something or precious samples, we're able to get a lot of data from a single individual. And so briefly, here's just some photos kind of this workflow. So I've been fortunate to get a lot of material, for example, from the Ice Age expeditions. We'll be collecting more in Antarctica and through these workshops. So I've got awesome students who have been going through and imaging literally thousands of specimens. These are a couple of my favorite photos. These were both taken by my postdoc, Carmen Cobo. So this is a Pro-Neomenia that she and my master student, Maddie, are working on. And I believe this is Eleutheromenia, but I like it because it has a mohawk. We can then do microcomputed tomography on those same specimens. And admittedly, I'm cheating here. This is not a meiofaunal animal. This is a very big Antarctic Neomenia. But micro-CT technology has continued to improve and be really high resolution. So we can get really nice high resolution images from even near meiofaunal size animals. And this is super valuable for seeing, for example, the reproductive anatomy. So you're just seeing one plane. We can rotate these specimens digitally, do histology through them, and see a lot of the structures we would otherwise see in histology. Now, I need to reiterate that we can't see everything we can see in histology. There's some things I just can't see, and so we're not getting as much data. But when we're done, we can just put this thing back on the shelf. It's totally harmless to the specimen. As I said, we can do scanning electron microscopy and generate really nice images like this. But aside from being kind of cool looking, these are taxonomically informative. And so just by looking at the sclerites of these different taxa, I can tell you this is the genus Virenia. I know the family of this one because it has these hooks, which are distinctive. This is one we know that has a mixture of scales and solid spines. And so this can be really informative. Now, as I said, sometimes they just look like little spiny worms. And so the sclerotome can be kind of hit or miss in terms of taxonomic information. We can then use those same specimens for DNA barcoding. And again, I've had some really wonderful undergraduates who have been very active in barcoding. We've been aiming to do at least one specimen of each morphospecies from the Ice Age material and the Antarctic material we have so far, and produce this absurdly large tree. And again, we have a nice photo and images from the SEM of almost every specimen we've processed in the lab. And so aside from just building a database for this, this has been really useful for addressing some questions about the biodiversity of the group. And so, for example, a former student in the lab, Tessa Hawkins, and now a new undergraduate who's continuing this work, Caroline Bond, is barcoding what I thought was one species of Irenia argentea from the northeastern Atlantic. And it turns out that we have on the order of like 8 or 9 cryptic species from a genus that was previously only described to have 2. And I say cryptic species, but now my student Megan Chungo, with some help from our postdoc Carmen Cobo, has been sectioning these and looking at their internal anatomy, and we'll be able to formally describe several new species. And so this barcoding has been kind of a hypothesis generating tool in that sense. And so using these DNA extractions, we can also do, for example, phylogenomics. So this is a tree from a paper we published last year, just kind of trying to reconstruct the higher level phylogeny of the Aplacophorans. But this is a group with on the order of 500 species and something like 25 families, and we've sampled maybe 20 species in less than half of those families. The problem is it's hard to get material suitable for transcriptomics, as we did here, for many of these groups. And so what we want to do is use our existing transcriptome data and some new genomes that I'll talk about to design probes for target capture phylogenomics. And so really this is where we take DNA, we make a Illumina sequencing library, we use probes to hybridize just the genes we care about and pull those out and just sequence that subset of the genome rather than the entire genome. And so this allows us to enrich these DNA libraries, as I said. And the great thing about this is we don't need RNA, we don't need high molecular weight DNA. And so even specimens that were bulk fixed in the field and maybe not treated well, or older museum material, can work for this. They can't be too old or they can't be even too mistreated, but we've been able to get material out of some specimens that had pretty shady looking DNA. Another nice thing about this is it does work well for meiofauna. So we've been using the NEB Next Ultra 2 FS kit, which can go down to as little as 100 picograms of DNA, which is pretty easy to get out of even some of the smallest meiofaunal organisms. And so this works really well for sampling Aplacophorans. And as a nice bonus, although we're designing probes for nuclear protein coding genes, mitochondrial DNA, of course, is really abundant relative to nuclear DNA, and genes like 18S and 28S are multi-copy. So we end up pulling those out with these probes anyway. And so we get kind of bycache of data that are compatible with other studies that have used these more traditional molecular markers. And so our data are kind of versatile as well. So to design good probes, we need to be able to use our transcriptome data, but then map those probes to a genome to identify those that are going to be matching to like an exon-intron boundary. And so for that reason and many others, my student Megan Chungo is sequencing not 1, but 5 Aplacophoran genomes. And so again, we're sort of cheating by starting with some of the really big ones, but so this is a giant one, Neomenia megatrapezoida from Antarctica, and another big tropical species, Epimenia babai. And Megan's been able to produce some really nice draft genomes for these using a combination of Oxford nanopore long read sequencing and Illumina sequencing. And we're hoping to add Hi-C data for these soon. And we're waiting for sequencing centers to open back up, but Megan has plans to sequence 3 additional species, 2 that are bona fide meiofauna, and 1 that's meiofauna-ish. But so to generate genomes spanning the diversity of this group, which will help us in our phylogenetic efforts, but also Megan is really interested in the biomineralization of these sclerites, these scales or spines that cover the bodies of the Aplacophorans. And so she hopes to do some Evo-Devo work and to look at the gene regulatory networks patterning these structures and compare that to what's known in shells and, for example, oysters and abalone and things like that. So I just want to end a little bit more broadly. We are very interested in mollusks, in particular Aplacophorans and chitons in the lab, but I often joke that I'm kind of taxonomically promiscuous, and so we have projects on all sorts of different taxa going on. And my student Nick Roberts in particular is really interested in the phylogeny of Lophotrochozoa, aka Spiralia. And so we're collaborating with Torsten Struck at the University of Oslo to sequence a number of Lophotrochozoan genomes to try to develop contamination-free references that could then be used to help decontaminate existing transcriptomes. And so I'm not going to go into too much detail about this, but if you have questions or would like to chat with Nick, I'd be happy to put you in touch. But in short, what we've been doing so far is generating transcriptomes from a lot of myofaunal animals. And so briefly, this is a fairly straightforward approach that pretty much any molecular biology lab can do. We're extracting RNA with this ambient RNaqueous microkit. We elute in basically as small of a volume as we can. So if you elute in about 15 microliters, you end up getting about 12, which gives you plenty for the 10.5 microliters that goes into this Takara SmartSeq HT kit. This is a new kit where you have your reverse transcription reaction and PCR amplification all in one tube. The only trouble we have is trying to guess how many cycles we need to do based on the animal. Usually it's about 16 to 18, depending on how many cells it has, how glandular it is, how much RNA it's making. And then we can make sequencing libraries, something like the Illumina Nextera kit or the Takara HT Plus kit, which is new, we haven't tried yet, has a built-in library prep as well. So we end up with a dual index Illumina sequencing library. And I will admit our failure rate is non-zero. Sometimes meiofauna presents some challenges, particularly when we tried initially just putting the animal directly in the tube. These kits are made for cell cultures. They say you can put cells directly in, but it seems that some of these invertebrates have inhibitors or things like that. But this works fairly reliably. And we've got a tweaked protocol that we're happy to share, and also happy to work with those on a collaborative basis if folks need help generating some transcriptomes for their critters of interest. And then lastly on that note, a problem that folks often encounter with meiofaunal genomics is we have to put the entire animal in the tube. We can't dissect a 100 micron long worm and get muscle tissue or something like that. And so we're getting not only the animal, but whatever it ate, whatever's growing on it, et cetera. And to help deal with problems like that, my former master's student, Felix Tallien, wrote a piece of software called PhyloPyPruner. And so this is for identifying orthologous sequences in transcriptome or gene model data, but it also has some built-in features to help identify exogenous contaminants in transcriptome data, which is really challenging, and get rid of those. And so that's up on GitLab for anyone who might be interested in that sort of thing. And so with that, just a shameless plug. As I said, Aplacophorans are kind of rare. And if you happen to come across any, feel free to pop those in RNA later ethanol for me, and I'll send you my FedEx number. And with that, thank you very much for tuning in today. I have to thank my awesome lab, a number of collaborators, and I'm sure I'm missing some, so apologies to those. And thank you for your attention. I'm happy to answer questions as long as folks have any.